All right, so now we're going to move on to the next unit, and this is going to cover chapters 4 and 5. So this lecture is going to talk about energy and enzymes, because the whole point of chapter 4 is how your cells obtain it. So first we need to define what is energy. So it's your ability to do work, or something to do work, not just you. And one important thing is energy is neither created or destroyed. It just goes through cycles. So it just transforms from one way to another. So the bond that we may have in some of the chemical bonds that we have in our food, in like glucose, for example, that will be transformed, use that to power ATP. Then ATP will help drive your muscles, and then that can lead to kinetic energy. Our example with light energy, the sun. So that when plants absorb the light energy, they go through the process of photosynthesis, and then they can actually use that to replenish their ATP sources and also make starch. So energy doesn't really just go away. It goes from just one state to another. Now, there are some different types. There's potential, which is the energy stored in bonds. This is the one we're really going to care about. You store it and you put it to use later on. Sometimes you have to make a bunch of bonds that you're going to need to break later on. And when we get more into cell respiration, you'll kind of see why that happens. And then the other type is kinetic energy, energy in motion and movement. But we're going to really focus on this chemical energy, the one in bonds. So we have to use energy pretty much to power everything. It's like the currency of the cells, you need to power your cells, and that's going to be done via ATP. Now, we have to consume living and non-living components of the environment to get energy, and we'll talk about how the consumption of these products then drives the production of ATP in the next lecture. So just a little information of how do we measure it. This is a measurement called calorie. Here's a technical term. It's the amount of energy required to raise the temperature of 1 gram of water from 14.5 degrees Celsius to 15.5. I don't know why they picked these specific temperatures, but that's just the way it is. We're used to thinking about calories, but actually when you see this, it's actually a kilocalorie. We measure it. Now, when it comes to the main macromolecules that we talked about the carbs, the protein, and fat that you'll consume through your diet. And each of those different macromolecules gives you a different amount of energy. So your 1 gram of carbs gives you about 4 kilocalories. 1 gram of protein gives you 4 kilocalories, and 1 gram of fat gives you 9 kilocalories. Now, excess calories that you don't use are going to be stored as fat. So that's why they kind of tell you to limit lipids and the fats and all that stuff, because it could lead to an excess of calories and the body just winds up storing. So here's your one slide on thermodynamics. I know it sounds really scary, but I just want to kind of introduce the two main laws. So the first law, the total amount of energy in the universe is constant. And I mentioned this on the very beginning. Energy is just changing from one to another. It's neither created or destroyed. We're just kind of transferring it from one place to another. And the second law, it's going to go from a more useful form to less useful form. And most of the time, we'll see this released as heat. But overall, the goal of this chapter is talk about how we can take something as simple as one little glucose molecule, remember that's a very simple monosaccharide, and use that to convert to make 32 ATP. So let's talk a little bit more about what ATP is. So the term is adenosine triphosphate. Now this is your energy currency of the cell. It's like the money that makes your strive and go. Now, how we're going to do that is we're going to use the chemical energy stored in food. We're going to break it down into smaller components. And as we do that, we will gain phosphates that we can use to power and recharge ATP. Now, the whole process of breaking down food into smaller products is called catabolism. So I always like to think of catastrophe. And if you remember, that's an exergonic reaction that's giving off energy. Now, the great thing about ATP, it's kind of like a rechargeable battery. So let me show you the parts first real quick. So this top one is ATP. You're going to have a sugar, and you have this base adenine right here. And you have 3 phosphates, 1, 2, 3. So adenosine triphosphate, 1, 2, 3. Where the energy is stored is in this bonds between the phosphates. So when we need a packet of energy, what we do is we harvest that bond right there, and now we have a molecule that's called ADP, adenosine di for 2-phosphate. Most of the energy is going to be in this first bond right here. Now, when we're at ADP status, we can actually use it one more time to get to AMP, but most of the time we just go and recharge it at that point. So metabolism is what we're really focusing on these 2 chapters. And it kind of talks about how your cell links all these different processes and ultimately to generate ATP. And you got to have all these processes working in sync, because if it gums down in one area, it's going to affect the other one. And we'll kind of talk about that a little bit more when we talk about fermentation. Now, you do get various sources of food. So you'll have your carbs, and we'll see how that goes through glycolysis, and then that will feed into the TCA cycle and then electron transport chain. But you also get fats in your diet, which can be stored as triglycerides, which will feed into the TCA cycle. And you also have amino acids from your proteins, and then that could feed into different parts too. Ultimately, all the different food sources do feed into one or more parts of overall metabolism, and we'll kind of hit on that a little bit later on too. So just a reminder, there's different types of reactions. There's going to be reactions that give energy and reactions that require energy. So we cannot hit on this before. If a reaction gives off energy, it's an exonic, or exiting energy. Energy is the product. If a reaction takes energy in order to form, it's called an endergonic reaction. So I explained this slide when we did chemistry, so if you're kind of confused, make sure you go back and look back over or watch that lecture. Now, what metabolism is going to do is we're going to couple reactions. So what we're going to do is we're going to link a bunch of reactions together in order to make it to our final goal. So what we're going to do is we're going to use catabolism to where we're going to break down some molecules, and we're going to get little energy packets from that. And then we're going to use that to fuel another step that is maybe where we build things up. Now, what we have to do is we need to harvest our little energy packets, and there's different ways that we're going to do that. One way is by gaining phosphates, and we can directly recharge ATP. Another way is we're going to pick up electrons, and this is going to be a big one that we'll talk about in the next lecture. But we're going to use these electrons, especially this NADH, a lot to really the electron transport chains, where we're going to get a lot of the payoff that's going to happen. But I want you to appreciate from this slide that the reactions do play off each other. So we might have an exergonic reaction and gain the energy from that one to kind of push the next reaction to happen. So if you've ever been bike out and where it's hilly, you'll probably use the energy that you gain from going downhill to kind of get your speed up. So when you see that hill that's coming up, you don't have to try as hard. So by using energy and transferring it where I got it here to help drive this next reaction, it's kind of like the overall goal that your cell wants to do. So how do we get the end? That's not working today. So we want to go from where we get one of our favorite things, mine's pasta. How do we generate energy? So that's what we're going to talk about, and mostly in the next lecture. But first, I need to mention how we're going to get there, and we're going to need some help. So this next part talks about enzymes. Now, what enzymes do, they're proteins, but they're a catalyst, and their job is to speed up reactions. I'm one of those people that tells you to work smarter, not harder. You know, there's a way to do something. If we were in lab, I will tell you the most efficient way to do it. I know where things are going to give you trouble. I'm trying to help you get through it. I am like your enzyme, your catalyst. Because if I just let you flounder there, it's really not really effective of your time or mine. So what they do is they speed up reactions, but there's a key thing. They're not used up. So they get to go and do that reaction over again with someone else. So I might have had you for the Tuesday section, and then the Thursday section, I'll do the same thing. I use this little guide like a little mountain climber to kind of explain it to you. So pretend you live way over here on the other side of the mountain, and every day you got to go home. So the way you know to do it is go over the mountain, boom, boom, boom. So you got to fight all the lions, tigers, and bears, wherever this may be. Now, what an enzyme is, think of him as like a mountain guide. He knows these mountains better than you do. You just know this is just one way to go. Now, what the enzyme does is he's like, hey, I know this is your goal. Boom, boom, that's a bad start. What he's going to do is he's going to tell you, he's like, I know a shortcut through the mountain to get you to your final goal. You're still going to have to put some effort to get there, fight Gollum, whatever you're going to occur. But he found a way for you to do it quicker and probably easier. So once he gets you through the other side, then he's going to go and find another lost person that's trying to help and get him across the mountain. That's what enzymes do. Now, when it comes to naming enzymes, there's certain things like, how do I recognize what it is? My key thing, look for anything that ends in ACE. If you see something that ends in ACE, it's probably a good indicator that it's an enzyme. And then most of the time, the prefix will tell you what it's going to do. So here's a table that kind of shows you some examples of what I'm talking about. Isomerase, isomers is going to help with rearranging. Lysase are going to be splitting things up. Proteases are right here, break down proteins. So think of protein, aces. Lipases break down lipids. Sometimes they build it up, sometimes they take it apart. Sometimes they're moving functional groups. Enzymes do a lot of different things. Now, some of them might have common names that break this ACE rule right here, but as longer in the field, you realize like, hey, I know trypsin is an enzyme. I use it in cell culture all the time. So there's a few parts of it that we're going to talk about, and how does it function? So what an enzyme does, ultimately, it's going to take what we call a substrate, so the initial thing we're starting with, and it's going to convert it into a product, so our final thing we're going to get. So this example right here, the enzyme is this gray blob right here, and their substrate is what we're starting with. So what's going to happen is the enzyme and the substrate are going to bind together. When they do that, they form what's called the enzyme-substrate complex. Now, once this binding happens, the magic happens here, but between the enzyme and the substrate. And it's going to convert this into a different product. The enzyme's not going to change. It's still going to stay the same, but we're changing the substrate into something else. After it's done making the change, it's going to release the products. Now, this enzyme is reusable, so then he's going to go back and for more substrate to do it over again. So here's where the magic happens. So sometimes I would use Pac-Man to talk about enzymes. So if you want to know where the magic is going to happen, it's called the active site. So the active site, if you want to think of it, is like Pac-Man's mouth, where the substrate is going to bind to the enzyme. So our active site is this little shaped thing here. Now, active sites are very specific. By that, I mean, which is a good thing because you don't want enzymes acting everywhere. You want to make sure they're going to target specifically what they're going to do. If it's a protease, you want to make sure it's only attacking a certain type of protein, because if not, it's going to attack every protein in your body, and that could be quite harmful. So I use the term the lock key model often for enzymes. The substrate has to fit exactly right into the active site and for the reaction to happen. So an example I would use is I have like keys, and sometimes multiple keys will fit in the same door, but unless it has the right specific ridges, you won't be able to turn and unlock the door. So it's one thing for it to just kind of slide in, but it's another thing for all the right functional groups to hit in the right places for the reaction to actually happen. So how does it work? I'll get a little bit technical. So we're going from reactants to products. In order to do this, it's going to take some energy. And the amount of energy that's going to happen be needed to make this happen is called what's called the activation energy. So that's shown in purple on this graph right here. So if we had this reaction happen on its own, which it's still, it's going to take this amount of energy, shown on purple. What an enzyme does is it makes it happen quicker and with less energy. So what it does is it lowers the activation energy. So if you compare with the red to the purple, you can see there's a considerable, maybe like a third, almost half the amount of energy is required for the reaction to happen. So it's still like with going back to my mountain guide. It's still going to take you some effort to crawl through the mountain, but not as much energy and effort as crawling over it. Now, the way that it lowers this activation energy is by affecting the transition state. Now, the transition state is what happens when we go from substrates to products, or we're converting it from one thing to something else. And this is that changing, that real awkward state that this substrate is going to be in. What it's going to do, it's going to make this transition state happen a lot easier. So it's like, if you want to think of teenagers and going through puberty, it's going to give you all advice stuff to help make it last, still going to last the same amount of time, but to make it a little bit easier. Less time is better when it comes to some transition states with some of these chemical reactions. So how do we do that transition state? Remember, we're going from, go back one thing to another. So there's going to be some chemical reactions that are going to be happening. Maybe we're making bonds and breaking bonds. So how does this enzyme? Sometimes it just does it in different ways. Sometimes you bring two substrates together in the proper orientation. So you think of it like a blind date. So you'll have substrate A and substrate B. They're just hanging out together, just doing their thing. But until you bring them right next to each other and get them in close proximity, they won't interact and form the final product. So maybe the enzyme is like that. You're the matchmaker. You're going to bring these two and set them up. And hopefully they're going to form the couple that you want. Sometimes you just provide the right chemical microenvironment. So it's like, hey, why don't you come here? It's like luring, I guess, a kid in a van with candy or something like that. You're going to butter up, persuasion, bring them in. Here's some idea conditions, come here and I will help this happen. You can pick some torque on it, be the strong arm. It's like sometimes, it just needs a little bit of effort to make the reaction happen or break that bond. Enzyme might do that. Sometimes you just got to rip the Band-Aid off. Maybe the enzyme's kind of like doing that situation. Or it could go and remove functional groups to or from the substrate. Carrot stick, it's like, here, you come here, give me that, and I'll give you that. It's kind of like a trade-off. Kind of works the same up with this buttering up and persuasion. So why should you care? Well, pharmaceutical drugs is a big thing of why you should care. Now, sometimes enzymes, they can be overactive. As I mentioned, they're reusable. They just do their job and they go back and look to release the products, go look for more substrate, do it over again. So they're reusable. That's an important thing. They're very specific, but sometimes, some active sites look the same. So that might create a problem. Or maybe they're too good at doing their job, so maybe you have to turn them on. Now, what we do is we use pharmaceutical drugs to kind of confuse them. So an example I'll give right here is Lipitor. What it's going to do is inhibit HMGA coenzyme A reductase. So this is a key enzyme in production of cholesterol. And we talked about cholesterol, how, having some is important. It's part of your plasma membrane. It has some other functions, but too much can be bad. Now, what this drug is doing, it's finding the enzyme HMG-CoA reductase, and the drug fits exactly into the active site. So remember that lock and key model. And it goes to the active site, and it's not going to make a product sometimes. Sometimes it's just going to keep the enzyme busy. So it's like if you gave him some chewing gum or something to keep his mouth busy and keep him quiet. It's like giving a toddler your phone, which you regret immediately. But they're quiet, you're giving them something to occupy them. That's how a lot of pharmaceutical drugs work. It's really kind of cool. Now that's one way, if we can do it synthetically. but the body regulates it also in different ways. One way is doing it through allosteric inhibition or activation. Now, on previous examples, I was showing you the active site was just already there looking for its substrate. Well, sometimes if we want to make sure the active site can't do its job, well, let's either block active site or let's change the shape. So if active sites are very specific, if you change the shape of it, then it can't bind a specific target. So there's one way that you can do that, either activate it or inhibit it. So we have our enzyme right here, shown in this gray half-moon circle, and it has an allosteric site. So allosteric means is we're going to cause a change in shape in another location. So right now, we're going to do inhibition. So we have a functional enzyme with its active site is that half moon right there. When the inhibitor binds to it, it causes a conformational change to the enzyme. Now the active site is not this half moon, it kind of folded in on itself. What's happening is Pac-Man's mouth has now changed shape and it cannot bind to a specific substrate, so you inhibit it. Then the body might recognize, okay, we got into the right levels. It can remove this inhibitor, and then enzyme can go back to its normal thing. Or sometimes you'll have enzymes that are permanently turned off, just waiting to be activated. So you need a certain signal, it binds to the allosteric site and makes it active. So these things go forward and back. It's like a feedback loop that your body will use. So form equals function. If you don't have the right shape, then you can't do your job. So one way then I talk about how we can make an enzyme not work as well is denaturation. So now I talked about the folding of the enzyme, how we get to that tertiary 3D shape. And that's pretty much shape is going to determine what it's going to do. Now, the active site is really specific. So if you change the active site, the enzyme can't do its job. Well, when we do that, we do the process called denaturation. Now, denaturation is when you pretty much take it from its 3D shape and kind of go back to that primary shape. So it's like unraveling it right here. At this point, this is useless. You have no active site. It can't do what it's going to do if it's an enzyme. Now, as I mentioned, I talked about this before. Sometimes this is reversible, sometimes not. The example I used was like if you cook an egg. Eggs have lots of protein. Now, when you add heat to it, what happens is it causes those bonds to break between different amino acid side groups, and it kind of takes on a different, form. You can't go back. You can remove the heat, but it's not going to go back to the original one. Now, sometimes if you remove the heat or put it in the right environment, it can. So here's a graph that kind of talks about it. Now, enzymes are very specific. They're reusable, but they like to work in a certain range. Kind of like us, you probably have an optimum temperature, lighting, and all that stuff that you work best at. Enzymes are the same way. So like this graph is showing you right here in the bottom. So we have rate of reaction here on the Y and temperature on the X. And then the red line is showing you the rate of reaction. At this, you can see the reaction rate is the best at optimal temperature, which is probably 37 degrees right here. So when you start going above it, you start to see the rate go down drastically. As temperature slowly going up, the reaction rate's getting better. So your money zone is right here. The same thing works for pH, Depending on what the enzyme is, it might like a different pH. So enzymes work in your mouth, probably like more of a neutral pH versus an enzyme that works in your stomach. It probably likes a really acidic pH. Environment's going to make a difference. So hopefully after this, you appreciate how we're going to use energy to generate ATP. We're going to be harvesting those bonds and coupling reactions to help make other bonds that we possibly need to break later on. But ultimately, metabolism is really complex. It's a lot of pathways interacting with each other. Now, one way that we're going to do this is we're going to use the help of enzymes to make these reactions happen a little bit faster. And with enzymes, just remember that they are reusable. They're very specific on what they're going to bind to, and they have an optimum range that they like to work in. So here's a few videos to watch about enzymes and energy if you're having a little bit of trouble. And then that is the end of this one.